Your brain has already decided whether you will remember this sentence. It made that decision before you finished reading it, not afterwards, before. Here is how you have felt that happen. You walked into the kitchen for something, and now you were standing there. The reason is gone. It was in your head 4 seconds ago. Now there is nothing where it was. Or this one. Someone tells you their name. You shake their hand, and by the time you let go, the name is gone. You have always been told this is a small failure, a lapse, a tired brain. It is not. Nothing leaked. Nothing slipped through a crack. Something inside your skull made a decision. It looked at that name and marked it for deletion. And that decision was already made before the name reached your ears. This is happening right now. It happened every second you were awake today, and every waking second of your life. It is happening to every person alive. 8 billion editors working without permission, deleting the day as it happens. If you're the kind of person who cannot let a sentence like that go, you are in the right place. Brain tenacity goes down these holes properly. Consider sticking around. And tell me in the comments what you lost today that you would have kept. Because here is the real question. Who is doing the choosing? On what criteria? It is not you. You have never once decided to forget a name. Somebody in there is deciding for you. For most of the last century, nobody could prove it. Then a lab in London caught it in the act. The year is 2006. At University College London, a researcher named Len Otten runs a simple experiment. Volunteers sit with electrodes laid across the scalp. A cue appears on a screen. Then, a moment later, a word. Then another cue. Another word. Hundreds of times. Later, they are tested on which words they can recollect. Otten was not interested in the words. She was interested in the cue. In the small gap before the word arrived. And this is what her team found. The brain activity during that gap predicted the outcome. Not the activity while reading the word, the activity before the word existed on the screen. Words preceded by one pattern were recollected. Words preceded by another were lost. The paper ran in Nature Neuroscience in 2006. The university's own announcement put it plainly. They could predict memory of an event before the event happened. Sit with that for a second. The screen was blank, the word had not been shown, and the fate of that memory was already leaning one way. There is an obvious objection here. Maybe that is just attention. Maybe the volunteers were concentrating harder on some trials. Preparation, not memory. It took 3 years to answer that. In 2009, a team led by Sebastian Guderian and Emre Düzel published in the Proceedings of the National Academy of Sciences. They worked at Otto von Guericke University in Magdeburg with University College London. This time, the tool was magnetoencephalography. It reads the faint magnetic fields your neurons throw off. 24 people, the same shape of experiment, and they measured theta waves, a slow rhythm around 4 to 8 cycles a second. In the moments before each word appeared, theta amplitude rose or fell. When it was high, the word was later recalled. When it was low, the word was lost. Every single one of the 24 showed it. And here is the part that kills the attention objection. It did not matter which task the volunteers were preparing for. The effect held regardless. This was not a person getting ready to concentrate. This was a brain state. The team traced the source to the medial temporal lobe, the part of your head that builds memories in the first place. So the gate is real. It sits upstream of the moment. Which leaves a harder question. What happens to the things that get through? You might assume they simply fade. That memory is a photograph left in a window, bleaching in the sun. That is the model most of us carry. It is wrong. In September 2019, the journal Science published a study from Nagoya University in Japan. The lead author was Shuntaro Izawa, working in the laboratory of Akihiro Yamanaka. Thomas Kilduff at SRI International in California worked with them. They were studying a group of cells deep in the mouse hypothalamus. Those cells produce something called melanin concentrating hormone, MCH neurons for short. And they send long fibers straight into the hippocampus, straight into the memory center. The team switched those cells on and off. Turned them on, and the mice got worse at remembering. Turned them off, and the mice got better. Better than normal. Removing the cells entirely improved memory. Which tells you something uncomfortable. Those cells were not helping. Their job was subtraction. And there was one more detail. The effect only appeared during REM-sleep. Switch the cells off while the mouse was awake, and nothing changed. Switch them off during other stages of sleep, and nothing changed. Only during the dreaming phase. Every night, a circuit switches on and begins removing. That is the honest reason you cannot hold on to a dream. It is not that the dream was flimsy. It is that something came for it. A word of caution here because it matters. Those were mice. Nobody has switched off these neurons in a human volunteer. The circuit exists in us. Whether it does exactly the same work is not yet settled. Now widen the lens. Because this is not only about last night. Try to remember being 2 years old. You cannot. Almost nobody can. Psychologists call it infantile amnesia. For a century, the explanation was that infants do not really form memories. That explanation is also wrong. Infants form memories perfectly well. They lose them. In May 2014, Science published work from Catherine Akers and Paul Frankland. They were at the Hospital for Sick Children in Toronto. Their subject was neurogenesis, the growing of brand new neurons in a region called the dentate gyrus. You do this your whole life. An infant does it at a furious rate, and each new neuron has to wire itself into an existing circuit, which means rearranging that circuit, which means damaging whatever was written on it. Akers and Frankland tested that directly. They raised neurogenesis in adult mice after those mice had formed a memory. The memory degraded. Then they did the reverse in infant mice. They slowed neurogenesis down. The infant memory survived. Then they went further. Guinea pigs and degus grow most of these neurons before birth. Their infants do not show infantile amnesia. So the team raised neurogenesis in those animals artificially. And infantile amnesia appeared. They induced it. Read that back slowly. Your first 2 years were not too faint to record. They were recorded. And then paved over by the growth of the very brain that was recording them. So the gate refuses most of it. The night crew removes more. New growth erases the earliest layer. Surely then, what is left is safe. The memories you have carried for 30 years. The ones you can still see in detail. Those are the survivors. Those are the archive. No. In August 2000, Nature published a paper by Kareem Nader, Glenn Schaaf, and Joseph Ledoux. They worked at New York University. It broke a rule everyone thought was fixed. The rule was this. A new memory is fragile for a few hours while proteins are built to hold it. After that, it is set. Nader tested whether it stayed set. He took rats carrying a fully consolidated fear memory, old in memory terms, locked in. He reminded them of it, just the reminder. Then he blocked protein synthesis in the amygdala. The memory did not survive. Recalling it had unlocked it. The act of remembering returned that memory to the same fragile state it was in on the day it formed. And there was a window. Block the proteins right after the reminder, and the memory is gone. Wait 6 hours, and nothing happens. By then, it has already been written back. This is called reconsolidation, and it means the thing you think of as looking something up is not looking anything up. It is opening a file with edit permissions. Which brings us to the uncomfortable part. Those were rats. Does human memory actually bend? There is an answer, and it is one of the most argued over findings in psychology. In 1995, Elizabeth Loftus and Jacqueline Pickrell published a study in Psychiatric Annals. They were at the University of Washington. It is known as the Lost in the Mall study. They recruited 24 people. From each person's relatives, they gathered 3 true childhood stories. Then they added a 4th. Getting lost in a shopping mall around age 5. Crying. Rescued by an older woman. It never happened, not to any of them. Across repeated interviews, roughly a quarter of the participants remembered it. Not agreed to it, remembered it. Some added details nobody had given them: what the woman looked like, what the shop smelled like. For decades, that study was attacked, and some of the attacks were fair. 24 people is a very small study. So in 2023, Gillian Murphy and colleagues ran it again, properly. Pre-registered. 123 participants. 35% produced the false memory, higher than the original. And here I have to be straight with you, because most videos on this topic will not be. That number is still contested. Andrews and Bruin reanalyzed the same data. They argued many of those reports were fragments of real events. Their estimate was closer to 4%. Other researchers, including Wade and colleagues, reject that reanalysis. The argument is live right now. Nobody watching this knows the final number. But notice what nobody is arguing about. Every side accepts the effect is real. They are haggling over the size. So we have an editor. It vets the moment before you have it. It deletes overnight. It paves over your childhood. And it revises the survivors on contact. Why? There are two serious answers, and they do not agree. The first answer is efficiency. In 2017, Blake Richards and Paul Franklin published a perspective in the journal Neurons. Their argument comes partly from machine learning. A system that memorizes every detail of its training does not become smarter, it becomes brittle. It overfits. It cannot handle anything it has not seen before. Forgetting, they argue, is not memory failing, it is memory working. The goal was never an accurate record. The goal was a good decision tomorrow in a world that has changed. The name you dropped genuinely was not worth the storage. The second answer is stranger. In 2000, Martin Conway and Christopher Pladell Pierce published a model in Psychological Review. They called it the self-memory system. In their account, an autobiographical memory is not retrieved, it is constructed, on demand, each time. And the thing doing the constructing is what they called the working self. Your current goals, your current beliefs about who you are. Which means the filter is not optimizing for truth, it is optimizing for coherence. It keeps what fits the story you are telling right now. It quietly loses what does not. Under the first theory, you are a well-designed machine. Under the second, you are a novelist who believes he is a historian. Both theories fit the evidence we have. Nobody has settled it. Let me make this yours. In the 1980s, a Dutch psychologist named Willem Wagenaar ran an experiment on himself. Every day, for 6 years, he recorded one event from his life. He logged 4 things about it. What happened, who was there, where, when. 2,400 events. Then he tested himself, giving himself some of those cues and trying to reproduce the rest. He published it in Cognitive Psychology in 1986. Now do the arithmetic. 6 years is about 2,190 days. Call it 16 waking hours a day. That is roughly 35,000 hours of being alive. 2 million minutes. And the total record, kept by a memory researcher who was actively trying, was 2,400 moments. Roughly 1 per day. Everything else was gone before he could write it down. And Wagenaar was cheating. He had a notebook. You do not. Whatever you did last Tuesday, most of it has already been processed. The gate refused it. The night crew swept it. And the version that survived has already been revised by you thinking about it. Now run that forward for 80 years. The thing you call your life is not a film you have been watching. It is a few thousand edited frames. There is one last turn, and it is the one that unsettles me most. Go and fetch your happiest memory. The clearest one you own. You just opened it with edit permissions. Nader's window is open right now. Whatever state you are in this evening is in contact with that memory while it is soft, tired, hopeful, lonely. And in a few hours it will be written back, slightly different. Here is the cruel geometry of that. The memory you treasure most is the one you have handled most, which makes it the one that has been rewritten most often. The clarity you feel is not evidence that it is accurate. It may be evidence of how many times it has been through the editor. You are not remembering your past. You are remembering the last time you remembered it. And the editor already decided which parts of today were worth keeping. It has finished with this video. Some of what you just heard is already marked. Whether any of it is still there tomorrow was never your call. Next in this series, we go after a stranger fault in the same machine. The moment your brain insists that something brand new has happened to you before. Déjà vu. Scientists can now trigger it on demand. I will see you there.